Hi everyone, welcome to my channel Pure Biology, and today I come with another video, that is the DNA repair. Okay, before going to this topic, I want to say that about DNA repair, already two topics related to DNA repair I have already uploaded and previously in my channel. Okay, so you just check out that all, and this is the, I think, third type of repair system. Okay, mismatch repair system. This is called the mismatch repair system. So here the mismatch system, that is the repair system. What is the function of this repair system? This can detect the mismatches and that occur in the DNA replication. That means during the DNA replications, if any mismatches are occurring, these are totally corrected by the repair system. And here the enzymes that functions as first of all, they recognize the mismatches, base pairs. Second, they determine which base in the mismatch is the incorrect one. And the third one is that excise the incorrect base and carry out the repair synthesis. Okay, there are three functions that are done by the enzymes. And now this repair must be made in the daughter polynucleotide. What is saying that when the newly strand or the new strand that is formed from the parental strand, then sometimes the mismatches formed or the error occur. And where this is occurred in the daughter polynucleotide. And here first it is possible to distinguish which one the parental DNA and DNA strand and which one is the newly formed DNA strand. And first, the repair system, they recognize the which strand is the newly formed strand. And here this is done by the presence of methyl group, because when the newly formed strand is occurred or it is formed from the parental DNA, so it can be distinguished easily by the presence of methyl group which is present in the parental strand. Okay, in that way the repair system or the enzyme can determine which one is the parental strand and which one is the newly formed strand. And during the period while the newly synthesized strand awaits the introduction of the methyl groups. Okay, this is the one thing that before mismatches or before determination of the mismatches or recognition of the mismatches, first of all, during replication, the newly formed strand, they awaits for introduction of the methyl groups, and that is possible or that is already present in the parental methyl group. So when this repair system occur from the just period of the replication, when the methyl groups that try to enter or try to attach with the newly formed DNA strand, this is the actual period of time when the mismatches totally recognized and corrected by the enzymes. Okay, so here one thing another is saying the E. coli DNA is methylated because of the activities of the DNA adenine. So methylation is possible for which for the activities of what? For the activities of the DNA adenine methylase. This is the enzyme. So methylation is possible due to the presence of enzyme that is called the DNA adenine methylase. Okay, and this converts the adenine group to the GNA methyl adenines. Okay, so what is the function of the DNA adenine methylase? This convert adenine group to the adenine, means ATGC. These are the base pairs, nitrogen bases that present in the DNA strand. So it converts the adenine to G methyl adenines. And which one is the sequence? The sequence is the 5' GATC 3'. And the DNA cytosine methylase, this is another enzyme. This converts the cytosine to 5-methyl cytosines. Okay, so here the two important thing possible, that is one DNA adenine methylase, this converts the adenine to methyl adenines, or another thing is that DNA cytosine methylase, this converts that cytosine to 5 prime, not 5 prime, 5 methyl cytosines. And here the base pair area or the strand region, this is the 5 prime CCAGG 3 prime, and another region is the 5 prime CCTGG 3 prime. These are the two regions where the conversion of the adenines and the cytosine is possible. The methylation is not here, the methylation is not mutagenic. It is not mutagenic, and the modified nucleotides that having the same base pairing properties as the unmodified versions. Okay, so here the properties of the base pair not be changes or not be separated. That means the modified DNA base pairs have the same properties like that unmodified DNA base pairs. And when these mismatches are corrected, there is a delay between the DNA replication and the methylation. Okay, replication and methylation, it must take a time. Okay, and within this time, the DNA repair system scan the mismatches and correct them. There are three mismatch repair system in E. coli. One is the long patch, another is the short patch, and the third one is that the very short patch. So here I only try to describe about the long patch and how the mismatch occurs here. And one important thing is that the three proteins, these are called the mu proteins. This coded the mu genes, these are involved in the correction of the in the mismatch repair system. And the three proteins are MUTH, MUTL and MUTS. Okay. So this is the mismatch repair system, and there is something I have to explain. So first I wrap this one or clean this one, and then I will explain about the last part. Okay, so here this is the rough diagram, and what is the role of MUT H and what S? Okay, so the MUT H protein that cleaves the unmethylated strand on the S side of the G in the, sorry, not S side, it's the 5 prime side of the G in the GATC sequence, and the combined action of the DNA helicase. Okay, here MUT H and MUT S both are involved in this cleaving process. MUT H and MUT S also. MUT S. The two proteins, these two proteins generally cleaves the unmethylated strand on the 5 prime side of the G. One second. So actually the MUTS and MUTH both proteins, these are actually functions combinedly. This cleaves the unmethylated strand at GATC sequence, and combined action of the helicase 2, DNA helicase 2 and the exonuclease 1 that removes the segment of the new strand between the cleavage site and the point just beyond the mismatch. Just beyond the where the mismatch is occurring, that point, beyond that point. That means which portion is cleaved? The segment of the new strand between the cleavage site and the point just beyond the mismatch. Okay, where the cleavage is occurring and where the mismatch is there, beyond the mismatch portion and the cleaved site, is total segment is removed by the exonuclease 1 and the helicase 2. Another two important enzyme, DNA polymerase and DNA ligase, they fill the gap and form the totally correct strand. Okay, correct DNA strand. So this is the diagram you just see here. One mismatch is seen just A and C both pairing with each other. This is the mismatch portion. First, MUTH and MUTS both bind with this site, and now MUTH and MUTH that cleaves the DNA. Okay, this mainly made the role of the MUTH is that the cleaving of the DNA. Okay, so here that cleaves the DNA, and the helicase 2 exonuclease 1 that removes the segment, and in the next part the DNA pol and the ligase excise and join the segment with the correct base pairs. Clear? So this is the mismatch repair system, and I here explain this topic is a very brief way. And in my next video, I will try to explain about the recombinational repair. So continue with my channel, and if you are new in my channel, obviously subscribe and share this channel.